My name is Noah Davidsohn, CSO and co-founder of Rejuvinate Bio. I want to thank ARM for having us. I'm very excited to talk about our novel approach to treating the diseases of aging. And so traditionally and historically, what you see in gene therapy is that they primarily go after single gene disorders. And I think what we want from the field generally is to go into more complex, larger patient populations and treat more patients generally. And one of our investors and members of our SAB, Dr. Catherine High, thinks Rejuvinate is uniquely poised to be a first mover into this space. The reason is because we operate at the intersection between aging and gene therapy, in which we take novel genes and are able to treat them to multiple age-related diseases. So as I mentioned, we are sourcing our genes from the aging field. What this means is that they have unprecedented safety profiles. They were discovered using transgenic mice in which they would overexpress these proteins for the entire life of the mouse. And the quote-unquote side effect of this overexpression is that they lived longer, healthier, with a reduced risk of getting the problems associated with aging like obesity, diabetes, cardiac enlargement, cognitive decline, et cetera, et cetera. The three genes that we're using are FGF21, TGF-beta1, and alpha-klotho. RJBO1 is our first offering and is a combination gene therapy using FGF21 and soluble TGF-beta receptor 2, which actually decreases TGF-beta1. FGF21 is primarily involved in metabolic processes, and TGF-beta1 primarily in fibrotic development. And alpha-klotho seems to be a magical cure-all that is effective in protecting cells throughout the body: cognitive, heart, kidney, muscle. So this is actually a sneaky pipeline slide because it's slightly different than your standard pipeline slide. What we're actually showing here is that RJBO1 is a platform in and of itself because it can be applied to numerous different age-related diseases. So you can see here that it can be applied to both the cardiovascular, metabolic, and renal space. Most important thing is that we will be looking for an indication that has a high unmet need with a large probability of success to get us into clinic and get our proof of concept data very quickly. We are looking to be in the clinic in 2023. Another unique thing about Rejuvinate Bio is that we are going forward further with our large animal data. Everyone has to do a preclinical large animal experiment in order to show safety and tox. What people don't realize is that's 90% of the way to getting an animal therapeutic approved. And because our therapies play across numerous different diseases that have similar underlying pathologies, we're able to apply this to other natural diseases that have high unmet need in the animal population. Specifically, our first indication for the animal health department is mitral valve disease. This not only provides a recurring revenue stream by partnering this out to another animal health player, but it provides us with a recurring data stream that feeds into proof of concept in the real world for our therapy and new treatment modality, where we're going after the diseases of aging with a more broad gene therapy. And so it's really exciting. We'll heretofore have patient numbers in the tens of thousands for our gene therapy entering into we'll be selling our gene therapy into this market in 2023 for dogs. So the approval is much faster, and it's just one step away from doing that IND-enabling large animal tox study. And as I keep mentioning, Rejuvinate Bio plays in a number of different spaces. One of those is the cardiac space. And our path for the cardiac space, like I said, we're looking for a disease with a high unmet need and a high probability of success. After talking to a number of different KOLs, we've decided that ARVC is an ideal candidate to go into. ARVC, arrhythmogenic right ventricular cardiomyopathy, is an orphan disease, and we'll be quickly able to get proof of concept data in this disease, which will show both the safety and the efficacy. And the efficacy has similar underlying similar underlying pathogenesis that would prove and point toward being able to expand into other indications that have a much larger prevalence, and these are potential pathways here for our secondary and tertiary indications and where we would be able to expand. Here we're showing the mouse model data that makes us believe we can play in the cardiac space. This was actually published in 2019. This is all the work I did in George Church's lab during my postdoc. So I won't belabor this slide since it's been around for a little while, but the main point here is that in an ascending aortic constriction model in mice, we're able to prevent heart failure using our therapies. In the left-hand side for fractional shortening, you can see the control mice continue to decrease in functionality. Fractional shortening is a measure of heart function. In the middle, we're seeing our ability to prevent remodeling in the heart. In the middle, you see a dilated phenotype for the control mice, and on the right, for our therapy-treated mice, it looks much more like a normal control mouse in which the chamber size is normal and the heart is pumping well. And on the right, we're seeing histopathology slices where we're seeing a marked decrease in fibrotic development, which is causing the remodeling in the middle that you can see in the echocardiograms. So while that is very exciting, it is a mouse model. And I think more exciting here is that we have disease in the real world in people's pets. We're currently running a pilot study through Tufts Veterinary School in which we are treating dogs with naturally occurring mitral valve disease. It has a similar underlying pathology in that it develops remodeling and fibrosis, eventually leading to heart failure and death. The current standard of care, pimobendan, is unable to significantly alter the trajectory of the disease, and when you're diagnosed with this disease, patients usually have about 2 years to live, unfortunately. And the first step in the disease remodeling process is, because the mitral valve has a lot of regurgitation, you end up getting a left atrium enlargement. And left atrium enlargement is directly correlated to an increased hazard ratio of moving successively through the prognosis of the disease, eventually ending in death. And so you normally would slowly get an increase in left atrium size. And what you see here is very impressive because we're able to actually reverse the atrium size with our therapy in combination with standard of care. So it's very exciting. This is highly suggestive that we're able to reverse disease dynamics and actually increase these patients' lives and hopefully stop the disease in its tracks. Along with the remodeling data I just showed, we also have heart function data in which the fractional shortening is increasing. That's the ability of the heart to pump the blood more. And so these two pieces of data are very exciting because it's highly suggestive that we're able to actually reverse the disease. And the naturally occurring nature of this disease is highly suggestive that it would be very relevant for a human population that has similar underlying pathology. As I keep mentioning and trying to hammer home, taking genes from the aging field allows us to play in numerous different disease areas. So the same RJB01 actually has data to support our going into the metabolic disease space. And similarly to the cardiac strategy, our goal is to find a patient population with a high unmet need, with a high probability of success, to quickly get us that proof of concept data that our therapy is both safe and effective, and that the primary outcome would suggest that we would be able to move into other indications with higher prevalence. And so we've chosen familial partial lipodystrophy after talking to a number of KOLs in the field that believe that our therapy would have a very good chance of serving this patient population and helping them. The data that supports us going into the metabolic space is in mice from that same paper that was published in 2019. This model is a high-fat diet model of obesity in which mice are overfed a high-fat diet. They become overweight. The dotted black line is the high-fat diet mice. The the green line on the bottom is a normal diet mouse, and you can see that line that connects the two is high-fat diet mice given our therapy, that remain on a high-fat diet. That's the key here, is that even though they're on a high-fat diet, they are still losing weight and becoming normal, lean mice. You might ask yourself, well, what if they're just not eating? That's not the case. In the supplemental of this paper, we show that they eat just as much, if not more. So it's very exciting that we're able to reverse obesity in these mice. The other metric that is very important here is insulin resistance. Obesity generally tends to lead itself to having insulin resistance, and these obese mice are no exception. HOMA-IR is a measurement of insulin resistance. A higher number is bad, and what you see with our therapy is we're able to completely reverse the insulin resistance and actually increase insulin sensitivity even beyond that of a normal diet mouse. And again, really exciting is the fact that we have data in dogs that corroborates our data in mouse models. So here we have non-obese beagles. These are just beagles free-fed in a lab environment. As anyone with dogs knows that dogs like to overeat, and these dogs are no exception. They generally eat a little bit more than they should, gain a little bit more weight than they should, and once given our therapy, are actually brought down to a normal lean body weight. And we have not seen a single negative adverse event from all the dogs we've injected. These are the pets that we've injected with mitral valve disease, the dogs in the lab that we've injected with our therapies, not a single adverse event. And that is what comes with these unprecedented safety profiles that we have from our aging genes. With that, I'd like to end talking about our team. My co-founder, Daniel Oliver, CEO, we've actually been friends for about 17 years. We met at Caltech. And George Church, my mentor, without him, I would not have had this opportunity to work in this space. He's been extremely supportive, and he has basically tripled down on us. He's given me the opportunity to work in his lab. He's invested in our company. He's a co-founder, and he's on our SAB. And finally, but not least, Kathy High, a investor and a member of our SAB. She has been extremely helpful with her advice and support, and we're eternally grateful for her continued advice and support. And with that, I can take any questions. I think I was actually quicker than I thought.